today's second presentation is Dr. Grant Poole. He is a product manager with the agrigenomics team, Thermo Fisher Scientific. Dr. Grant received his PhD from Washington State University in 2010 in plant pathology and wheat genetics utilizing molecular techniques in breeding for disease resistance and has spent over 8 years in various corporates like Bayer, Syngenta, developing crop varieties utilizing molecular-based techniques in mapping and genomic associations with major traits of economic importance and has involved in a release of several successful crop variety products launches throughout the western region of the USA. So let's listen to Dr. Grant Poole and his insightful presentation, and then we'll move on to the next part of our webinar.
Is Grant Poole, and I'm a product manager in agrigenomics at Thermo Fisher Scientific, and I am here to present to you about our microarray, Axiom microarray technology, and I will touch on our AgriSeq NGS technology as well, but I really want to focus on our Axiom microarray catalog offerings for wheat, corn, and soybeans, three of the largest worldwide crops. And in this presentation, I really just want to cover a little bit about who we are, our technologies, and go into our catalog offerings for wheat, corn, and soybeans in the array area, as well as some of the partnerships that we've been able to form over the last few years, as that's really what we do at Thermo Fisher Scientific, is we partner with our customers, whether they're in public or private institutions, to help meet their genotyping needs and help us develop our technologies as well. One of the things that we really focus on at Thermo Fisher is our mission, and that is to enable our customers to make the world a healthier, cleaner, and safer place. That's what we embody as employees at Thermo Fisher Scientific. When you look at our offerings, they're quite diverse, and if you're not familiar with some of these technologies, it can be quite daunting to try to figure out how these are working and operating. But when I was a graduate student 15 years ago, I can tell you these technologies allow you to genotype crop genomes much quicker than we could then. Okay, so starting with our microarray technology, this is our highest resolution technology, where we can look at hundreds of thousands of SNPs that have been discovered over the last few decades and start to figure out which ones are important in certain breeding backgrounds. And then from there, we can move down to a 384 format, down to 10 to 50,000 markers of interest. The 384 is much higher throughput in terms of sample number. And then once you characterize your 10 to 50,000 markers, a lot of times customers will come to us and they'll have 2,000 to 3,000 markers of interest. And at this point, we direct them to our AgriSeq targeted sequencing platforms on our Ion Torrent sequencer. And then from there, customers often get down into where they have been operating in the capillary electrophoresis and TACMAN assays for a good number of years. And these are the economic markers of interest. These are in the tens to less than 100 markers. These are markers that have economic value that every breeder needs to have in their germplasm. So really, the left hand of this schematic is what has been developed in the last 10 to 15 years, whereas CE and TACMAN have been around a long time. So again, what I want to focus on in this talk is catalog offerings, and this is actually all the catalog offerings that we have available on the website, from plants and crops to livestock to aquaculture and companion animals, and then model organisms and also the microbiome. And we have over 70 catalog designs that are available. We have multi-species designs that are available as well in both the 96 format and the 384 format. The 96 format is the higher density, hundreds of thousands of markers. The 384 format holds 10 to 50,000 markers. And these can all be purchased on our website. at thermofisher.com. We are in the process of always looking for cooperators to develop new catalog designs. So we partner a lot of times with consortias and academic institutions to do that kind of work. But when we approach customers, or customers approach us, they usually approach us with these questions, right? So they may have a marker number in mind, they may not, but they certainly probably have a sample number in mind that they want to analyze. They want to look at making selections in their population. So they have a sample number in mind. If they don't have a marker number in mind, they can go to our Axiom array platforms and look at hundreds of thousands of SNPs. In our catalog designs, they can order those on the website and start to figure out which ones are important in their breeding backgrounds, and then get it down to the tens of thousands or even few thousand, and go to our sequencing, targeted sequencing technologies. But we're usually working with the customer to balance all of these, and we design both arrays on the hundreds of thousands of marker side and panels in AgriSeq with a custom design process. And there's an AgriSeq team that will lead our customers through a custom design process, and an array team that will do the same. And we do this with cost in mind to the customer. So at the end of the day, we want to develop genotyping solutions that are fast, affordable, robust, and user-friendly. So how is this applied, really? And my background has been breeding for the past 15 years. And I can tell you that breeding involves a lot of different things, but usually, traditionally, prior to markers, it took 12 to 15 years to release a wheat variety, for instance, into the market. And here is a breeding schematic on the development of a typical wheat variety. So again, it can take a lot of years, but really it's that year 3 to year 6 where breeders are looking for making selections. They want to get the number of individuals from 20,000 down to 400. And that's where genomic selection, marker-assisted selection, comes into play to make those selections and increase efficiency, reduce development cost, and really develop a durable product. I've been in the breeding industry in the private side for the past 8 years, and I can tell you that there's been a number of times when I've seen genetic resistance break down. This is a picture of a wheat plant that we had basically developed over the course of time, and we were bringing it to market. We'd invested a lot of money, a lot of time, and a lot of resources into developing this variety. Then the stripe rust race shifted, and there was only one single gene that was responsible for the resistance in this, and we really didn't know what it was. We were using passive breeding to bring this to market, and the genetic resistance broke down really quick. So I think that's where our technologies really can come in and allow our customers to make intentional breeding decisions, intentional genomics and selection and genomic prediction, and really develop a durable product. And there are economic diseases for each crop that I'm going to cover today, but there's really those economic traits of interest that our technologies can help our customers find multigenic sources of resistance, or just mine genomic areas for certain types of traits that they're interested in. This is an example of our Axiom 35K wheat array that was used to bring in genetic resistance into wheat. And so this 35K was used to develop a genetic map and identify genetic resistance that was then introgressed into wheat. And you can see the power of this technology. And these authors basically reported that this array is more suitable for gene mining and target breeding compared to the Illumina 9K and 90K iSelect. They found that those Illumina arrays did not have a good coverage of the D genome. One of the things that you really should be aware of when you're genotyping in crops is there's a different level of ploidy across organisms and different chromosome numbers. So we have a staff of trained bioinformaticians or bioinformatics scientists that can help you navigate this, both on the design side when we're designing our arrays or our AgriSeq panels, but also on the analysis side. So I put this example in there. This is a wheat example of a genome map that I made in my PhD, and it shows you the diversity of wheat. So there's 21 haploid chromosomes here, and there's 7 sets of chromosomes and 3 different genomes. So you can see that wheat as a hexaploid is very dynamic. But when I was in graduate school 15 years ago, or 12 years ago, 15 years ago now, we were basically looking at single marker analysis, and we were finding single marker regions in the genome that we can use for breeding purposes to breed for certain traits of interest. Now we've come so far because we can look at so many different markers throughout the genome. So this illustration really is basically represents 40,000 SNPs that was correlated with phenotypic data or phenotype data. In this case, it was wheat and use quality data. And what these modeling approaches do is basically end up at a genomic prediction value. And so using machine and deep learning to drive these prediction models, and so with all of this resolution, with all these markers, it's very powerful. You can arrive at genetic estimated breeding values and different ways to look at the genome that are very powerful. In the lower right hand corner, this is an example of where I looked at 300 individuals in a wheat breeding population for stripe rust resistance, and found really quickly that the resistance we had in our breeding program was on chromosome 2A, 4B, and 7B. And then I was able to go in and see what resistance genes are on those chromosomes. I'm going to focus the rest of this talk on our array platforms and really our catalog offerings in maize, soybean, and wheat. So $51 billion in maize. It's a very high value crop, $25 billion in soybeans, and $9.3 billion in wheat. But wheat is a very staple food source in the world, very important world crop. Our maize genotyping array consists of a little over 600,000 SNPs, and it was developed by a public breeding consortium called Amazing, and involved the German Ministry of Education and Research. And it basically used a lot of 30 different inbred lines from international germplasm, as well as land races, elite lines, and hybrids, even teosinte lines. And the applications in the literature have ranged from GWAS and haplotype structure in maize to QTL mapping and LD analysis. This is an example of a paper that was published where this array was used. Basically 159,000 out of the 600,000 were found useful in this maize or this corn population. That was basically used to correlate sweetness, which is an end-use quality, to markers on chromosome 3. Those markers were identified, and single marker analysis was developed, and a TACMAN assay was developed out of that work. So this is how our technology can be used to develop single marker analysis. We also have a soybean array. This one is 180,000 markers. 63% of the SNPs were in gene-rich regions when this array was designed, and it was designed on the basis of wild and cultivated soybeans on an international level. One of the applications with a group in Korea basically found 170,000 useful SNPs out of the 180,000 SNPs and correlated that in a GWAS, genomic association exercise, with earliness, the earliness trait. So again, use the high resolution markers to find traits of interest and markers of interest very quickly. We also have partnered with the University of Bristol, Keith Edwards, to develop a high-density wheat array on this 96 format of over 820,000 markers, and Keith basically identified 35,000 markers from that and developed this wheat breeders array. There's also been scientists in France that have developed a breed wheat array, and you can go onto the website and see those arrays as well. This is just a summary of researchers that used the 819K arrays in blue. You can see the 21 chromosomes, and the 35K is the red dots, and you can see they basically summarize that there was equidistant coverage across the genome with both arrays, and that there's future utility of this 35K breeders array. So go onto our website and look for those arrays and check them out. So basically, you can see that we have partnered with public and private institutions throughout the world, and we are here to partner with you. Please reach out to myself or our technical sales staff located throughout the globe with any questions or interests that you have. Thank you for your time.
Thank you, Dr. Grant. It was really great to understand how your work from your research and your graduation times and how has that made an impact, and how great technology we have to offer to our customers working on plant genotyping.